So this question is asking us, well, it's telling us that in each human genome there are 3.2 times 10 to the 9 base pairs. So there's 3.2 billion base pairs in each haploid genome. Okay. Now if we think about what a genome is, we have a picture of a cell here, and the cell, if you blow up its nucleus, well, look into its nucleus, there's chromosomes there, and if you zoom into its chromosomes, this is made of really highly coiled up DNA. It's coiled up around these proteins called histones. But then if you really boil down to the molecular structure of DNA, over here we have this, which is the double helix, a very familiar double helix. And this double helix is made up of these base pairs. Okay, so each one of these base pairs, okay, well, rather 3.2 billion of these base pairs make up the entire haploid genome of a human cell. Okay, so this is the haploid number, and because human cells are diploid, it means that there's about 6.4 billion of these base pairs inside a diploid cell. Okay, so that's what the question is trying to say here. Now, the other information we're given about these base pairs in the question is that each base pair weighs 660. Now, this is a different way of saying things. All it means is that one of these base pairs, okay, the molecular weight of one of these base pairs is 660. Okay, the molecular weight often is expressed in grams per mole or atomic mass units if you're a physicist. And so it's just saying one of these base pairs, either an A and a T or a G and a C, on average the molecular weight on average is 660 grams per mole. Okay, so using this information, let's try figure out, or I guess how much a genome weighs, or how many moles there are in a particular genome. Okay, so the first step here, 3.2 times 10 to the 9 base pairs. Okay, so one haploid genome has 3.2 times 10 to the 9 base pairs. Okay, it's normally abbreviated BP. Now, if we know that the molecular weight of a base pair is 660, this means that 1 mole of base pairs is equivalent to 660 grams. So literally, if in my hand I'm holding 660 grams of base pairs, then in my hand I'm holding 1 mole of base pairs. So we can continue this conversion and we'll say, well, 1 millimole of base pairs is equal to 660 milligrams, and so on. But then how many millimoles of base pairs do I actually have in a haploid genome? Okay, and the way to think about this is, let's think about it in terms of what a dozen means. Okay, so if I have 12 of something, then you can immediately tell me that is 1 dozen. Okay, if I have 24 of something, you can immediately tell me that is 2 dozen. So the way that we're doing this calculation with dozen is obviously with 24. We're doing 24 divided by 12, and 12 is the counter for dozen, and that equals 2 dozen. So in a similar way, we have a counter for molecules, and that is the mole. Okay, just like there is the dozen, which is 12. Okay, the mole is 6.02 times 10 to the 23, which is Avogadro's number. So just like you can have 1 dozen eggs, you technically can have 1 mole of eggs, I guess. This just means you have 602 sextillion eggs. Okay, so it's a very, very, very big number, this idea of a mole. Now, if you've got, say, 6.02 times 10 to the 23 of something, okay, and I need you to tell me, well, how many moles is this equal to? Well, just like if I had 24 of something, to tell me how many dozen this equals to, you divide it by 12. If I have 6.02 times 10 to the 23 of something, you just have to divide this by Avogadro's number, which is the counter for the mole, and this will tell you that I have 1 mole of something. Okay, so if I have 12.04 times 10 to the 23 of something, to tell me how many moles there are in this, it's essentially divide by Avogadro's number, and this equals 2 moles. Okay. So we know now that this Avogadro's number can be used as a counter. Now, when we use it as a counter, we can now then express 3.2 times 10 to the 9 base pairs in terms of how many moles there are. Okay. So if in 1 mole there's 6.02 6.02 times 10 to the 23 of something. And this number here is just 3 billion, which is much smaller than over here, which is 6.02 sextillion. Then obviously the number of moles of base pairs in a haploid genome is going to be much less than 1. Okay. In order to work it out, what we do is we can, just like we did here with 12.4 divided by the counter, we can do 3.2 times 10 to the 9 divided by the counter, which is 6.02 times 10 to the 23. Okay, and so this, if we plug it into our calculator, okay, so this is equal to 5.31 times 10 to the minus 15 moles. Okay, the division we've done here is essentially the division that we've done over here. Over here we end up with 2 moles. Here we've ended up with 5.31 times 10 to the minus 15 moles. Okay, so now let's convert this into a prefix unit. Let's put something in front of here in order to get rid of the 10 to the minus. Now our prefixes start out with milli, and then they go micro, nano, pico, femto, followed by atto. So milli is 10 to the minus 3, this is 10 to the minus 6, 10 to the minus 9, 10 to the minus 12, 10 to the minus 15, and 10 to the minus 18. So over here, 10 to the minus 15, this means it is 5.31 femtomoles. Okay, so this is 5.31 femtomoles in a haploid, or femtomoles of base pairs in a haploid genome. Okay, so 5.31 femtomoles, what does this actually weigh? Well, we know over here that 1 millimole is equivalent, or weighs 660 milligrams because of the molecular mass of, on average, of a base pair. Okay, so if 1 millimole weighs 660 milligrams, then we keep converting down, and we eventually get to a statement where 1 femtomole is equal to 660 femtograms. Therefore, 5.31 femtomoles is equal to 3,504.6 femtograms. Okay, so this number of moles, 5.31 femtomoles of base pairs, is equivalent to 3,504.6 femtograms. Okay, 3,504.6 femtograms is equal to 3.5. If you go further up, it goes from femto to pico, and so this would be picograms. Okay, so in a cell, in a haploid cell, okay, if this cell was haploid, all the DNA in here in this haploid cell would literally weigh approximately 3.5 picograms. Okay, in a diploid cell, this would weigh about 7 picograms. Okay, so if you had a cell which is diploid, in that cell, all the DNA inside its nucleus, in a human cell, it would weigh about 7 picograms, which is quite amazing when you think about it. Okay, so now we have an expression that the how much DNA is there in each diploid cell? Well, that amount of DNA is 7 picograms approximately. Now the next part of this question asks, if a technique can detect specific sequences and it requires 0.075 nanograms DNA, how many copies of this sequence would this amount of human DNA contain? Well, let's again write up our units. And from before, we know that one diploid cell has about 7 picograms of DNA, and a haploid cell has about 3.5 picograms of DNA. This technique over here can detect something as long as it has 0.075 nanograms of DNA. Okay, 0.075 nanograms, if we convert it to a nice number, this is going to be 75 picograms. Okay, so now the question is, well, if you've got a technique which requires 75 picograms of DNA to work, and each haploid genome okay, carries around 3.5 picograms of DNA, then how many of these haploid genomes will be able to supply the necessary 75 picograms in order for this technique to work well? If one haploid genome or weighs 3.5 picograms, then let's put 75 on this side, okay, and then convert that. And that will turn out to be 21.4. Okay, 21.4 haploid genomes, or haploid human genomes, will provide enough DNA for this technique to work. Okay, so here we go. Technique to detect how many copies. The answer to how many copies is essentially 21.4 copies of the haploid sequence.